Okay, so good morning, everyone, and welcome to the 26th Keio Medical Science Prize lecture. So I am Haruhiko Shiomi, chair of the Keio Medical Science Prize Committee. So as we already announced before, the Keio Medical Science Prize in 2021 was jointly awarded to Professor Osamu Nureki and Professor Katalin Karikó. And today, I am very pleased that Professor Karikó will give us a special lecture on developing messenger RNA for therapy. So, on behalf of the committee, I would like to welcome Professor Karikó, though it's a kind of pity we do not welcome her in person. So the lecture will be chaired by Professor Nakahara, one of the committee members. So I am looking very much forward to the lecture. So Nakahara-san, will you please start chairing the lecture? Thank you.
Well, thank you, Professor Shiomi, and good morning, good evening to Professor Karikó. And thank you for attending the 26th Keio Medical Science Prize lecture. And I'm Professor Jun Nakahara, committee member of Keio Medical Science Prize. Today, it is my great honor to welcome Professor Katalin Karikó, the 2021 laureate for the Keio Medical Science Prize, to give us a precious lecture on her discovery of specific RNA modification leading to mRNA vaccine development. Now, please allow me to introduce Professor Katalin Karikó. Professor Karikó obtained Bachelor of Science in Biology and Doctor of Philosophy in Biochemistry from University of Szeged, Hungary in 1978 and 1982, respectively. She started her research career in Hungarian Academy of Sciences and moved to United States in 1985. After working at Temple University and Uniformed Services University of the Health Sciences, she joined the faculty as a research assistant professor at University of Pennsylvania. She was promoted to senior research investigator and then adjunct associate professor before moving to BioNTech RNA Pharmaceuticals as a vice president in 2013. Professor Karikó is currently a senior vice president of BioNTech SE in Germany, as well as adjunct professor at University of Pennsylvania. She has been awarded with numerous prestigious international prizes, including the 26th Keio Medical Science Prize in 2021 for her discovery of specific RNA modification leading to mRNA vaccine development, of which achievement led to the stunningly successful COVID-19 vaccines. So Professor Karikó, if you would kindly grace us with a lecture, please.
Thank you very much, Professor Nakahara, for the nice introduction. It is a great honor to receive the 2021 Keio Medical Science Prize, along with Osamu Nureki. I hope that I will meet you and him one day, maybe this year. And it is a privilege to belong to all of those scientists who already received this award. And I am delighted to be here tonight or for you this morning and talk about the RNA, because this is our favorite topic. And let me share my screen here. And oops. Oops. So, sorry. Is it okay? Can you see?
Yes.
Yes.
Thank
okay.
You.
So that today I will talk about how the mRNA was developed for therapy, and I took a picture of my medal, and this is right next to me here. I am very proud, and thank you very much. And so developing the mRNA for therapies didn't happen overnight when some people thought, but actually it was happened for many, many years or decades. And the RNA was discovered in 1961, and these are the major milestones which I think that were important for development, which took for 60 years to have finally an mRNA product FDA approved, which all of you know about, the FDA approved COVID-19 LNP RNA vaccine. So what are these major milestones? For me, it was 1978 when messenger RNA was isolated and injected to mammalian cells, and the encoded protein could be detected. 1984, when first time mRNA could be synthesized in a tube, and 1990, when it was injected into a mice and the mRNA coded protein could be detected. 2000, when we discovered that RNA was inflammatory, and 2005 we could come up with a solution to make it non-immunogenic, and the first publications came out in 2017 demonstrating that lipid nanoparticle formulated modified RNA. So I remember most of these things, except in 1961, I was just 6 years old. So I thought that maybe I would just show how because I was not coming from out of nowhere, just like the vaccine did not. So I thought that I would say something about my background. So I was growing up in Hungary. I was born there, and a simple household. My father was butcher, my mother was bookkeeper, and I have an older sister, and we had the usual childhood, you know, we were curious, we had the yard and we had animals around, and I had, my parents supported us to study well, and I had excellent teachers. So in elementary school, so what happened is that I was 13 years old, I already competed in biology competition, and just because Friday was the Women in Science Day, I thought that I will show you this picture when we were in 13 years old. All of these kids were biology competition winners, and later became chemists, biologists, physicians, and here I am when I was 13 years old. So after I had great teachers, and I went to the local high school, excellent teachers. Again, this is a small town, 10,000 people live there. And so to get from here to the universities was not as easy because, you know, only 20 people were accepted in a year to be biologists. But thanks to my good teachers, finally I made it. And in 1973, I was accepted to University of Szeged. Szeged is in the southern part of Hungary. And so I studied here from 73 to 78. And I selected this to go to this university because it had in Szeged also had this biological research center just open. And then I thought that that was my dream to one day to work in this institute. And so when I was a student, I already was interested, and when a student visited this research center and asked the professor that whether I can work there in summer. And he told me that, oh, you have the rest of your life to work in laboratory, go and go to fishery, spend the summer in a fishery institute where I collected a sample from fish and the fat part, and that was, and I also collected like little Daphnia and others, and took back and analyzed in the laboratory and try to identify that how the different chow material, what the fish are eating, how it changed the fat content. When I went back to the laboratory in the fall, Professor Farkas was the head of this lipid laboratory, and two of the colleagues Eva and Erno, they were scientists there, and they came to us that they need phospholipids, and because they are planning to make liposome. They already did some liposome and wrapped up the nuclei and delivered inside the cell. And so we had to look it up, protocol, and this was actually published in 1942, how to isolate phospholipids from cow brain. Being a daughter of a butcher, so it was easy for me to have in, and together we did deliver the plasmid into mammalian cell using the liposome. I am showing this was because it was the very beginning, and for every researcher or scientist or student, you know, the first thing what they do is so important. And it was important for me because with the rest of my life, I was thinking about this experiment because we could detect the encoded protein. And of course, it was with DNA and not the RNA, but after I finished and graduated in '78, I joined the RNA laboratory led by Janu Thomas. And the day I joined, I already learned from him that RNA has a cap because he synthesized, he's an organic chemist, and he synthesized the cap analog, and he sent it to New Jersey, where Professor Furuichi, who is with, in the panel today, and together with Aaron Shatkin, they try to identify the cap structure of the RNA, and they needed reference material. So all of my colleagues were organic chemists, Janos Ludwig as well, and then our role was to synthesize 2', 5' oligoadenylate. This molecule was just discovered the prior year, 77, in Ian Kerr in London, and this molecule was supposed to be an antiviral compound, and we needed very much some kind of compound with antiviral effect. The mechanism was that naturally in our body, the oligoadenylate synthase activated by double-stranded RNA can activate RNase L, and this RNase L will cut up single-stranded RNA, mostly viral RNA, degrade, and finally it stopped viral replication. So this was our work, and I used adenosine analog, actually a cordycepin, which is a 3' deoxyadenosine, to synthesize these compounds, and we did progress very well, but we ran out of fund, and so I had to leave and went to the United States. Here, Professor Suhadolnik's lab is also interested in this 2', 5' link molecule, but he was also interested in all kinds of modified nucleosides, especially which inhibit cell proliferation. And what was interesting is that the problem with delivering 2', 5' molecules was a problem. So eventually, when we participated in a clinical trial, we did not use these 2', 5' linked oligonucleotides to inhibit HIV virus, but we used double-stranded RNA. And this was a mismatched double-stranded RNA that we injected to the patient suffering from HIV. And these are, 1986, most of the HIV patients were dying. And unfortunately, this double-stranded RNA did not help them to induce interferon because their interferon system was compromised. So this is where, I stand here in 1987, whereas in the field of the who are working with mRNA, we started all the way back to the 50s when they tried to figure out that what could be the molecule that taking the information from the nucleus, from where the DNA was, to the ribosome where the protein synthesis is occurring. And of course, they looked at there, and the ribosome had an RNA, and at the beginning they thought that maybe that's it, maybe that's which is coming out from the nuclei and taking there. But it was an interesting experiment done by Spirin and colleagues in Russia. And what they did, they analyzed the DNA from different bacteria, and the GC content was very different of the DNA. And when they isolated the RNA, which the total RNA usually is ribosomal RNA, what they found this was identical. So they realized this RNA is not reflecting what was in the DNA, so probably it is not the messenger RNA. So they still had to look further, and other scientists did that in 1961, published in 2 papers, consecutive paper in Nature. So there is we know already that from the DNA, there is an RNA called messenger RNA is described. And as I mentioned here in this 2 paper, both of them mentioned that it was unstable. It is very short-lived molecule, and then the protein is synthesized from it. So the first, they discovered the RNA existence, but they couldn't synthesize. What they could, the scientists at that time, they could isolate. And what they used is reticulocyte, which is a precursor of the red blood cells, and they enriched for beta hemoglobin chain, and then they isolated that, and then they translated first in cell lysate or injected to frog oocyte, and they could detect the encoded protein. So these were the first RNA delivery experiments. And but the following year, in 1978, when the first time they used liposome to wrap up this RNA, which they isolated again from the reticulocyte, and they could fuse primary mammalian cells, which was mouse lymphocytes, and they could detect the protein. So again, 2 consecutive paper was published in summer of 1978, showing that mRNA delivered to primary mammalian cells can be translated to the encoded product. So some people were working on this field, tried to deliver the RNA, the isolated RNA, while other researchers, like one of them, Professor Furuichi, who worked in Japan, and he was the first one in 1975, reported that something interesting and a blocked structure is in the beginning of 5' end of the messenger RNA. On that summer, later on in New Jersey and also in Australia, other scientists also identified some kind of, they call bizarre 5' end. And this was eventually shown here. This is this Cat1 structure present in the 5' end of this RNA. And these are 1975, several Nature papers reported. Probably in 1975 was the year of the CAP, because by the end of the year in December, Bernard Moss' colleague in NIH, they demonstrated that the vaccinia virus has an enzyme which can put this kind of structure to the end of the viral RNA. So this was an important finding, and of course, even today, which is very important, what kind of CAP structure we are incorporating to the RNA. And after 1975, the next important milestone was 1984, when mRNA was synthesized in vitro. So these were previously mentioned, those were isolated RNA, but now that scientists could synthesize, and these were Douglas Melton and Paul Krieg in the Harvard University, who come up with that using phage RNA polymerases, sp6 RNA polymerase. The following year, it was T7 RNA polymerase from the phage. And these require small, short promoter, and the fidelity was very good. Prior to them, prior 84, other scientists already tried to also synthesize RNA using bacterial enzymes and others, but this was not so successful. So what they did, they cloned human beta interferon into the plasmid, and then they generated RNA. The RNA has the coding sequence, has the 5', 3' UTR. The cap structure they did enzymatically, they added, Bernard Moss gave them this enzyme, and then they injected this messenger RNA they made in the tube and to frog oocyte. And then they cultured the injected frog oocyte, collected the medium, and they demonstrated that the human interferon, which was secreted by these frog oocytes, were functional, inhibited the virus. It is also important to mention you here that this was in an early time, and the scientists shared all of their resources. And I myself have received this plasmid, which contains the promoter from Paul Krieg. At that time, there were no material transfer agreements signed. People just sent in an envelope to fellow colleagues. And a big step was helping the cell culture related experiments when Phil Feldner introduced lipofectin. Again, might mention that this was the time when scientists, one day they just published lipofectin, and the next day you just could purchase from some small company. That was the same for RNA isolation when the trizol was introduced by Chomsky. So the lipofectin is a positively charged lipid, and because the nucleic acid is negatively charged, so it helps to protect the RNA in tissue culture and then helps to deliver it inside the mammalian cells. In 1989, Malone, Feldner, and Verma published that they delivered messenger RNA using the lipofectin into cells. So it was very similar what was done in 1978, but now that it was not liposome, but actually lipofectin was used, and not isolated RNA, but this was in vitro transcribed RNA was used. 1990, when mRNA was delivered into a mouse, and this was John Wolf and colleagues did this very simple experiment. In vitro transcribed RNA coding for luciferase were injected to the mouse muscle. It was a very simple experiment, but a lot of people were inspired by this finding. And now that started the experiment moving from the culture cells to more like in vivo experiment. And these were the major publication. I have to say that usually in that time, in the early 90s, when I already started working with RNA, every year we might have one paper using messenger RNA, and not like today is maybe every hour publication out. So in 1992, vasopressin mRNA was used by Floyd Bloom and colleagues in a Scripps clinic, and they could actually treat this rat, which had a type of diabetes, and with vasopressin mRNA injected naked RNA to the brain. So it was not formulated with lipofectin or liposome, just injected naked. And French scientists, Martinon, Maulien, they were the first one who tried to use messenger RNA for vaccination, for infectious disease. And they did a liposome, similarly what I have done with this film method, and they delivered this to mice. This was 93. 94, in Lillström colleagues in Karolinska, they did the self-amplifying RNA naked injected, and this again, both of them for against influenza. But that was about all what was done in the vaccine field, because after that, most of the experiments were done for cancer vaccines. So first in Conry Curiel, that Alabama, they did vaccinate, again using naked RNA for tumor antigen. And most importantly, in 96, Eli Gilboa at Duke University, who did isolated human dendritic cells and treated them, and then injected back actually to the patient. So he did all the way to human trial, and Merix was the first company in 1997, which was established for human trial. And they did isolated dendritic cells from patient, and then they injected back after they exposed to the tumor-specific RNA, try to vaccinate. Unfortunately, it was not very successful, and couldn't be approved for clinic. So while the scientists were working in vivo, I was working in the cardiology at University of Pennsylvania, and using the T7 promoter polymerase, and generated urokinase receptor encoding RNA. We were interested in intima thickening and blood coagulation, and why I'm showing this example here, Elliot Barnett, and was a cardiologist. And again, all of my life, I worked with people who were expert in something else, not I had to do. So here I work with physicians, and we try to come up together with some treatment. Why it was interesting? Because this receptor, to be functional, had to be fully modified with a glycosylated GPI anchored to the membrane, and somehow delivering the RNA. All of this happened because we could detect that delivering the RNA resulted functional receptor. And so at that point, we knew that the RNA would be good for something, but we didn't know. And as Elliot had to leave, I was helped with David Langer, who worked as a resident neurosurgery, and he convinced the chairman that neurosurgery need molecular biology. So this was the bench I worked for 17 years. And together we started to figure out what the patient would need. And that was like nitric oxide synthase, mRNA for subarachnoid cause vasoconstriction to release it, and other enzymes. We went that time was thinking that the stroke may be causing problem because it's oxidative damage. So we did a lot, and constantly we improved the RNA, more protein was made, and we were very successful. But then David had to leave also, and I was left there, and that I met Drew Weissman in 1997, and he was interested to develop a vaccine for HIV. And as we met, I offered him that I will make RNA, and this is a HIV-specific GAG coding sequence he gave me, and then I inserted it to a plasmid, made the mRNA, and he transfected in human dendritic cells. That was just discovered at that time. What happened is that he was very happy because all of these markers were activated in dendritic cells, and a lot of GAP protein was made. But when identified all of these inflammatory factors, you know, I was very much worried that because I want to use this for therapy. And so we started to think that why this activation by the messenger RNA, which I thought that it is the same as inside the cell, why this would cause inflammation? Could it be that even if we injure our own cells, that inflammation occurs? And so maybe every time when you an RNA gets from outside and activate some dendritic cells, because it is abnormal for the RNA to be outside. So to find out that whether the cells which is inside the cell is the same way inflammatory like the RNA I am making, so we isolated the RNA from different compartments of the cells, and then we exposed those to monocyte-derived dendritic cells using lipofectin, and we measured TNF-alpha. And here in the purple, you see that the in vitro transcribed RNA induced a lot of TNF-alpha. Those which we isolated was not as immunogenic, but surprisingly, this tRNA was not inflammatory at all. No TNF-alpha was induced. Knowing that the tRNA has a lot of nucleoside modification, we start to suspect that, oh, maybe these are contributing that to this non-inflammatory nature of that RNA. And we're thinking that how we could make RNA which has this kind of modification. And messenger RNA has a lot of modification, and I'm showing you here this picture just to scare you a little bit that how many different modifications are there. And these were known, but what was not known is that which are the enzymes that introduce into the RNA. So we couldn't pick up the phone and order some enzyme and put to our RNA and try to see that whether it is less inflammatory. And of course, not all of them present, but most of them is present in the mammalian and eukaryotic cells. So that we try to figure out that if the enzymes are not known, the only option is that if we purchase maybe the triphosphate derivatives of these modified nucleosides, maybe we can try to incorporate them as a modified one to the RNA, because naturally we always make our RNA from the 4 basic nucleotides, and then post-transcriptionally those enzymes, which we at that time was not known, even today most of them are not known, they changing and introducing different modifications. So we decided that we have to maybe purchase it. I might mention here that at that time, we are in 2002, we already knew that naturally, actually, how the pseudouridine and 2'-methylation, so pseudouridine is here, how they are incorporating, how it occurring in the RNA. And this was discovered by my fellow scientist from Hungary, Tamás Kis, and I called him up and asked for an enzyme, but you know, turned out that those need special structure, and I didn't have those in my mRNA. So eventually, what we decided that we had to purchase modified nucleoside triphosphate and try to see that whether we can incorporate. We insisted that we just can purchase those, and we will use those only, which is normally present in the human body. The reason was that because in 1993 at NIH trial, 5 patients out of the 15 volunteers died due to the modified nucleosides, which is naturally not present in the body. This was developed to hepatitis treatment, but they were surprised that why those people died, because this compound was not toxic for not the mice and not the rat, not even for the monkeys. And it took more than 10 years to identify the reason why the human is so sensitive for certain unnatural nucleosides, because the human has a nucleoside transporter, which in the coding sequence has a mitochondrial localization signal. And what it meant that when they delivered these unnatural nucleosides, it pumped all of them into the mitochondria, and the patient died from acidosis. So we insisted that we are not using anything foreign, so that we just purchased these 10. This was only available nucleoside triphosphate, modified nucleoside, and then we could synthesize, use only 5 of them incorporated by T7 RNA polymerase. So we get these RNA, and those, every position, what instead of uridine, now it is pseudouridine was there, or 5-methylcytidine, or M6A, and so on. And now that we want to know that whether these are activating something, why we tested out toll-like receptors with the stable transformed cells, because we are in 2002 at that time about, and it was already known TOS3 can activate it by double-stranded RNA. It was discovered and reported 2001, and we did all of this. And why we tested out, we found that conventional RNA, which we synthesize without modified nucleosides, these are the blue one, those are activating and inducing high level of IL-8 in this transformed cell line. But when the TOL8 was present, you know, but when we had the nucleoside modified one, those were not activated. So we come and discovered this about 2004, and that was the same year when Professor Akira discovered and published in Science that toll-like receptor 7 and toll-like receptor 8 are sensing single-stranded RNA, and that's what we found also. But of course, we already knew that how to make it non-inflammatory. So when we tested out now the same set of RNA, which we generated, which contained a modified nucleoside on isolated primary dendritic cells, what we found is that some of the modified nucleoside containing RNA still was inflammatory, but some group was not. And when we looked at closer, we could see that as long as the uridine was modified, whether to pseudouridine, 5-methyluridine, or 2-thiouridine, those were not inflammatory and did not induce any TNF-alpha. So somehow nature selected uridine to be recognized. Of course, for us to develop some kind of vaccine, we were also, our product, mRNA, which code for therapeutic protein, which was always in my goal when I work in cardiology and neurosurgery. It was important whether they are translatable, and that's what we found, that not only it translated as well as the RNA which had no modified nucleosides, but it was the pseudouridine containing translated 10 times more efficiently. And so we published this in 2005 in Immunity, and we were, or at least Drew Weissman, my colleague, said that, you know, people were calling us to give presentation, but the only call came from Japan when in 2006 I was invited to participate in this symposium in Hokkaido. Here I am in the first row here, and my colleague who just joined at that time to the lab, Hiromi Muramatsu, is here in the back, and we participated here on this meeting. Professor Seiya invited me, and I presented the data from what I received with the modified nucleosides. So when we went back to the lab with Hiromi, we started to work on how to purify, and together with Drew Weissman, how to purify the RNA, because at the beginning we could still see that there were double-stranded RNA present, and we could also demonstrate whether it was U-containing or nucleoside-modified containing RNA. We could see and detect on a dot plot using the double-stranded RNA-specific monoclonal antibody, and but an HPLC purification method we introduced, we could eliminate completely the double-stranded RNA. It was very much improved the translation of RNA. Here in the blue, you can see that the translation level when it was delivered to primary human dendritic cells, that we get a very high level, several log more protein was produced by especially the primary cells. And so eventually, we had RNA, which was non-immunogenic, which was not containing double-stranded RNA, so it translated super well. And then we started with Hiromi to evaluate this kind of RNA in mice. We used here transit to deliver and complex, because the lipofectin was helping in the culture cells, but those were very toxic in vivo, so we used different formulation. And when we tested out, we made a erythropoietin coding mRNA, whether it was pseudouridine or uridine containing. And what we found is when pseudouridine containing RNA was injected to the mice, we could detect the protein, the encoded protein, for several days. And when we could see that it was functional, because single injection of RNA resulted hematocrit increase. The erythropoietin is responsible for red blood cell production. And we also demonstrated that when we injected once a week a very small amount, 0.1 microgram RNA to the mice, we could maintain the high hematocrit. Of course, most important was that such RNA was not immunogenic, because we could not detect any interferon. So this was where we stand. And again, when we get all of this data, we get an invitation from Takeda. There was a subgroup of New Frontier Science, and we visited Japan. I was there second time in Yokohama, and in the Takeda Research Center. And here we are with Drew Weissman. Very rarely we were taken picture in anywhere, and not in the laboratory, but here it seems that in Yokohama we have a picture when we are dining here. And unfortunately, we couldn't make a deal, because I even offered that I will go there and I will work and try to make it and help the RNA to get to the clinic. But when in 2013, we finally decided that here is the time to move and try to make and help nucleoside modified RNA enter to the clinic, we selected BioNTech to go there. And together with Hiromi Muramatsu, my colleague, we went there like on a mission that BioNTech in 2012 already had an RNA in clinic. So we thought that this would be the best company to go. And maybe somehow, maybe in our lifetime, we will see that the nucleoside modified RNA gets to the some patient and helping them. And this BioNTech is primarily oncology company, so we focused on those kind of studies. And with Hiromi, we spent most of the time trying to do everything which we have done in University of Pennsylvania, but now that we have to make it scalable, industrial level scale, and then the HPLC was not scalable, so we had to introduce other purification method and so on. And so we did a lot of time spent with optimizing the RNA. Those means that the wild type RNA can be codon optimized, the untranslated regions can be further improved. We did a Cap1 structure, which tremendously increased the translation relative to the Cap0 structure, where methylated 2' methylation is missing. And when we did all of these optimization and purification and modification, we could have an RNA which was translated very well, and long, very high level of protein, extended time could be detected. I just mentioned here one thing about codon optimization, which not for everything is optimal, because when some scientists and some approaches trying to optimize in a way that the codon optimized, that changing the codons and by this way eliminating uridine, which is the immunogenic part. Of course, we are replacing uridine with pseudouridine, so make it non-immunogenic. But you can also eliminate the uridine by changing codon and make it RNA GC rich. One problem with this one is that you still cannot eliminate all of the uridine, because there are amino acids which need at least 1 or even 2 uridine, and if your protein is enriched for those amino acids, then your RNA will be more immunogenic. In addition, codon optimization, especially for vaccine for infectious disease, could result elimination of out of frame important epitopes. It was just recently shown for SARS-CoV-2, or it can create an out of frame cryptic epitope which is unneeded. While we were working with Hiromi, my colleague, Norbert Pardy, who took over our old lab, he, together with Drew Weissman, were working on using lipid nanoparticle formulated modified RNA with a different experiment for vaccination. And here we all published together in 2017, when messenger RNA containing 1-methylpseudouridine containing 1-methylpseudouridine, and it was coded for Zika virus specific membrane protein. And when these vaccines were injected to monkeys, very small amount, 50 microgram was sufficient to protect the monkeys when it was challenged with the viral Zika virus. Here you can see that without with a control, very high level of Zika viral RNA was present, but when it was vaccinated, even the lowest dose, it was protected. So this led to the development, of course, similarly other laboratories published data like the Moderna. And but for BioNTech, of course, you are more familiar with this kind of scheme, which went on as January. We learned about the sequence information for the SARS-CoV-2, and developing the vaccine happened in less than a year. And by August last year, it was fully approved by the FDA. And so I don't want to talk too much about, because in these days, most of the people are expert on this. And then you could listen every day, maybe in the television, we learned that when the phase 1 trial, what was phase 2-3 trial ended. But many people thought that, this vaccine was come out of, just born in such a short period of time. And that was the first mRNA which went to human. But many people did not know that there are already messenger RNA was in clinical development. The most advanced is for heart failure. This started, the first patient was injected actually 4 years ago. And this was borne out on an experiment performed at Harvard with Lior Zhangyi and Kenneth Chen. And they injected VEGF-A mRNA to the failing heart, and then they demonstrated that naked RNA could improve the heart performance. It was also run a clinical trial for diabetic patient necrotic wound healing. And what was interesting is that this trial went on and entered phase 2, and the result was recently reported, and showing that here is an experiment when clinical trial 30 place, they injected the VEGF-A into the heart, and the patient heart improved the performance. These experiments were done during the elective bypass surgery, coronary artery bypass surgery. And in January 2022 was reported that the trial will move on and it will be entering phase 3. And other success with nucleoside modified RNA was also reported last year when Cas9 mRNA treatment was used for patients with amyloidosis, transthyretin amyloidosis, and this was Intellia was reported. This is a terrible disease when the patients are synthesizing the toxic protein, and what was the Intellia is that they had messenger RNA delivered in a lipid nanoparticle, and this was coding for Cas9 mRNA, and also co-delivered a guide RNA to the patient. And when the RNA, the messenger RNA, was translated in the cytoplasma, formed this structure which had a complex with this guide RNA, and had a nuclear localization signal enter to the nuclei and interrupted the genes which was responsible for the toxic protein production. So, and another success with the nucleoside modified RNA was for when we performed intratumoral injection of mRNA. These were recently published the animal studies, and where we injected the messenger RNA coding for these cytokines, and those can be used in melanoma and other head and neck cancer. But here this animal model injected to the tumor, and we demonstrated that the cells which know that immune cells which migrated to the tumor and made the cold tumor hot, they could circulate and eliminate lung metastasis. And this is going on in a clinical trial, human trial with Sanofi. And more experiments are ongoing in clinical level. Almost every week you can hear about vaccination, of course, which is not only now preclinical because the HIV already entered, yeah, and flu also in human trial. Different companies in Moderna as well, and a messenger RNA was used for induced tolerization for multiple sclerosis. This was published by scientists at the BioNTech, and where the mRNA coded for the autoantigen. And at this time, it was this particle did not contain any adjuvant activity, and injected IV to the mice, and the translation occurred in the spleen. And we could show two different multiple sclerosis animal model to cure of that disease. Genome editing is also ongoing, treating edema using VEGF-C mRNA to form new blood vessel, and And 2 weeks ago, Drew Weissman and colleagues here at University of Pennsylvania demonstrated that how they could generate CAR T cells in vivo in an animal model and treat a fibrotic heart with this one. So there are many experiments are ongoing, and it was already preclinical studies in animal studies were ongoing for many different disease models. But we knew about this who attended every year from 2013 to the messenger RNA conference because this was a small group of people who were focusing on, but as it expanded, not only academic but clinical researcher joined in, and regulators, and we educated each other. So this is how it all stands, and now I just would like to say thanks to all of my colleagues and who helped me on this journey. And this was a long and winding road to develop the mRNA for therapy. And I thought that maybe one more thing, I will show you one more picture, because for scientists who, maybe those women who had to, maybe they think that they had to make a decision that whether they have to choose between having family and having a child or a career. I can say that here is my daughter. She's a 2-time Olympic champion, and I could do all of this research and she could do her stuff. And so with that, I would like to say thank you for your attention.
Thank you. Thank you very much for the meaningful and valuable lecture. So now let us proceed to Q&A session. The audience, if you have any questions, comments, please press the raise hand button at the bottom of your screen. In Japanese, it is Kyoshu button. In your setting of the Zoom, and the moderator will pick up and unmute the questioner by one, and you can ask the question by yourself. Your video will not appear on the webinar, and if you have difficulty to speak in English, you can also use the chat box to ask the questions. So while waiting for questions, I would like to ask myself a question myself. Well, I have one comment and question. My comment is that as a neurologist specialized in multiple sclerosis, I was happy to see MS on your screen. And there's increasing evidences, especially in the beginning of this year, in Nature or other papers suggesting that the Epstein-Barr virus, EB virus, infection is associated with development of multiple sclerosis. And so I would just hope that you would consider raising a vaccine for that purpose, or maybe your company. And my question is that I learned that you had a long, story, but I was very inspired to see that you already had a strong desire to become a biologist in the future, ever since you were in the elementary school. That was very great. And was it your curiosity in biology which brought you here, or your willingness to contribute to medicine? Or in other words, at which point of your career did you anticipate that your science or scientific discovery will contribute substantially to the medicine? Do you have any answer to that?
I didn't know that. I think that every child is curious, and we had chickens, and I watched how it's coming out from the egg, and we had animals around the house. We had plants, and then we had our little garden. We put the seed in, and we watch how it grows. I think that all of the child is curious, and if nurturing teachers are there and the parents, then we will remain curious for the rest of our life. And I was 16 years old. I was just back to my home city, and they showed me that it was written in the book there that I said in 16 years old that I would be a scientist. I have never seen one. I just imagined that go there and figure out and will help somebody. And although I did always basic science, I always thought about from day one that, oh, it will be good for something. And that I was thinking about that. And of course, coming to the US, it was different. Here, I did not mention that I also established a company. So I worked in academic, work Hungary, work Germany, work US. I established the company. I worked for company like BioNTech was like 100 people, what now is 2000. When we went there, we were just actually in the campus of university and try to navigate there. So we learned, and as an immigrant like Hiromi and me, you know, you learned also adapt more quickly because, you know, things are so different.
Well, thank you so much. It's very wonderful to know that it's the curiosity that you drove, you know. Yeah, it's very important for young researchers listening to this lecture. I have one raising hand in my screen. Is it Mr. Miss? I'm sorry, Isolu my pass. Can you come up?
Hello.
Yes, please.
Can you hear me?
Yes, I can.
Yeah. Thank you very much, Dr. Karikó. It was an amazing and inspiring lecture. And I also want to congratulate you on well-deserved prize. And we as like regular people are like nowadays kind of getting benefits of this tremendous work. So thank you very much. I have a question, obviously, regarding the mRNA therapy. So we already learned that the mRNA can be used as a therapy, to treat all these diseases and improve the health. And my question is kind of more towards like a future research. What's your opinion on using the mRNA therapy, or like in general using the RNA for improving the let's say the lifespan of the people, or like let's say renew the tissue or improve the organs? What do you think about such therapy in the future? Thank you.
Yeah, so I mean, that's what I was actually always wanted to do. And then it was interesting because, you know, I went there, BioNTech, as I had 30 proposals probably that what I wanted to do. And, but, you know, in the small company, we had to always, somebody offer some money to, then we had to do what they wanted. And so we could, of course, you know, that you can improve the function of organ. We could see that it was done already for the heart. And for me, it was also, and for Drew Weissman, my colleague, important that the RNA is easy to make. And it is so affordable because you don't have to figure out how to purify. The body will make the medicine. And the other thing is we did not invent the RNA, the nature invented. Bacteria has the messenger RNA, we have the messenger RNA. We're just using, you know, this, and when we discovered that pseudouridine is, you know, incorporated, it took 10 years later that they discovered actually messenger RNA has pseudouridine. And pseudouridine is the fifth most abundant nucleoside in our body. So, the importance is that very simple to make. So if there is a factory, you can make a, you know, RNA for a vaccine. It can make for therapeutic purposes and others. And of course, we need more of you guys there to test out and having knowledge in certain area of health and organs or whatnot. Then to come up with ideas, and then you deliver the RNA, and then you can eliminate, you know, some kind of microbiomes, or maybe you are introducing some therapeutic protein which is not present there and or not enough, and so it is just endless. You know that this whole thing is a platform, and the limitation is the delivery, because you cannot deliver, and the RNA, no matter how you wrap it up, it won't go through layers of cells to your desired cells. Usually, you know, the surface, like epithelia, will pick it up. Of course, when it is secreted protein, like I showed you, erythropoietin and other, it didn't have to go, you know, with a big needle all the way to the kidney because normally kidney cells are making EPO. It was enough even, you know, sub-Q or intradermally, wherever we injected, the protein which was produced, it was modified. You know that erythropoietin actually 50% is glycosylated, so it heavily modified, but it was different cells could perform this process. So you have to think about that the mRNA is not, of course, not the drug, but the encoded protein, or what we have done in the neurosurgery when we try to make nitric oxide synthase encoding RNA. Actually, the RNA code for an enzyme which generates a gas, which can locally, just half-life is the gas is millisecond, the nitric oxide, but it can dilate the blood vessel locally, and that was our idea, for example. So it is your imagination that what it can be used for, and it is very simple to make.
Oh, yes, I think, yeah, it's very inspiring that you use the tool just to make your organism to work for you. So yeah, thank you so much. Thank you.
You are very welcome.
Okay, I see some raising hand, but before that, I have one question on the chat. I have one question from Dr. Matthias Wolf in the OIST. Is intramuscular injected mRNA only expressed locally for the COVID vaccine? Is the spike protein secreted or membrane bound? Do you know the answer for
membrane bound? Yeah, membrane bound.
Correct.
Yeah.
And I have some raising hands now. May I go to Mr. Yoshiyuki Kiguchi?
Thank you. Today is thank you very much for your excellent lecture. My name is Yoshiyuki Kiguchi, and I'm a clinical researcher in endoscopic division of the Department of Gastroenterology. So I'd like to ask you two questions about the major concept of medical research. First, what was the biggest failure in your research life so far? And secondly, how did you overcome the failure? And please, thank you.
Failure.
Yeah, mistake.
Yeah, failure. I mean, you know, all of the scientists has to be a good to come over failure, because you know that's what we most of the time. I don't say that the experiments are not working, because I had in a big sign up there, which was from Leonardo, that experiments, you know, is always work. Actually, your expectation which has the problem, because you didn't do that experiment, you expected the result for, so the experiment never fails. So we are failing because we are expecting something else. So a lot of you know, challenges were there, you know, how to do, you are to constantly do something, you know, we have to make modified RNA. How the hell we make it? You know, nobody has done. Should we do that? Where is the enzyme? No enzyme. Calling up colleague. So I mentioned that I call up my ex-colleague because he discovered how to put pseudouridine. So what I can recommend to you guys is make sure that with your fellow student or colleagues, make a good relation, because one day you will need their advice and follow what they are doing. Because I knew that Tamás discovered the pseudouridylation. Actually, I was there in a conference that he got a standing ovation because he finally discovered what the SNO RNA, the small nucleolar RNA, is doing. And so it is important, and also was important, I call up my other colleague from Hungary, János Ludwig, who we were a graduate student together, because I didn't know where the hell I can buy modified nucleoside containing triphosphate, you know, so that you need advice, so that I can tell you that make sure that you know what your colleagues are doing, or your fellow students follow them. So the failure is what I did not mention. When I was in high school, I also read a book written by Hans Scheje, who was Hungarian. They translated that book. And it helped me to, because I am not had any religious upbringing, so it helped me to guide me through life. And this, he coined the word, this guy in the 30s, stress. And it was said that the stress can kill you, but really not the stress, but how you perceive. And he said that you have to learn to handle the stress. So coming back to your, failure question. And so he also said that without stress, life is meaningless. You don't get up in the morning. Why you would get? You need stress, but those are positive ones. So excitement, anticipation. So he said that you have to learn to, the failure, kind of the bad stress, like you are fired tomorrow, and that will be a big stress. But if you focus on what you can do, okay, if you are already get, the pink sheet that you have to leave, don't spend any time, agonizing on it, but focus on that what you can do, and find a new job where you can, I wouldn't be here today if I would not be fired a couple of times in my life. So every time the failure, which other people perceive failure, it might take you to the next step. I never wanted to come to America. I never dreamed about that. I just, lost my job, and then I had to take a deep breath and coming here. So the failure, what you have to handle with is that try to see that some positivity, so that again, when my grant was rejected, after all of them, I am here. I never get an NIH grant for the basic R01 research. I get one for my company, but never get anything. And I was just keep writing, because I learned when I was writing, and it was that was counted. And when criticized, I also take it as a positive criticism. So not that, oh, these people are stupid, they don't understand what I am talking about. No, because it won't help me. But reading it and realizing, yes, I need to show that the RNA is making more protein. I didn't explain well. I have to do more data, generate data. So, and then it means that you can improve yourself. So Yoshiyuki. So
Yoshi.
that's what you have to do.
Please call me Yoshi. Yeah.
Yoshi.
Yoshi.
So that's what you have to do. That the failure, there is no failure. It is just you perceive that, that you get kicked out. That, and if you take it that, oh, okay, now that I have an opportunity to find the other job, and that you take it positive, that's not failure anymore. You get a new opportunity. One door closed, other opens.
Yeah, Sam, very thank you. Now I get very biggest knowledge from biggest, very greatest researcher, Karikó. Thank you.
Now you are waiting for you get fired, yeah?
Well, thank you so much. Now move on to the next questionnaire. Mr. Zen Tamura, are you with us?
Yes. Hi, my name is Zen. I'm a second-year medical student here at Keio Medical School. Thank you so much for your lecture. So my question is about industry and academia. So you talked a bit about your time at BioNTech and how you optimize the vaccine there. Traditionally, I think academia has been the driver for research, but with your experience in both academia and industry, how do you view industry's role in advancing science as opposed to academia? Thank you.
Yes, so that I was surprised that, and I'm so delighted to work at BioNTech, because what happened is that no longer was important, how thick your CV is, how many committee membership you have, which was counted very well in the academic area, more publication, just more, and nobody reads that, but you have to publish more. And there, who cares? You have to have a product which is functional. And this is what's so uplifting and delighting, that no, we need something which effective, because otherwise we can close the door and go home. Yeah, so that was one. The other was in academia is the colleagues are more in a competitive. Everybody needs to get the grant, so they have to be first or last author, and even they didn't started the work already, they are fighting over that. But there, whose name? Nobody cares. Who nobody asks who developed that drug or something. What everybody had to work together. because we are in together. And so that was also delighted that first of all, we have to have some meaningful goal, and the other is that everybody works together. So that was what was, of course, less, you know, if you want to do what you want to do. So as I mentioned, I had many ideas and I wanted, but I realized also that if I wanted to do it, I have to make my own company. And that's what I can recommend to you also, that you have an idea, don't try to, you know, go to different companies, please do that. No, make your own company. And you are the best who you understand, because you always wanted to do that. And I am sure that sooner or later, small, you know, venture capitalists will see the ideas good and they invest. There are RNA companies in Japan, like Akurna. They were there for like more than 5 years. You know, they were there in the RNA meeting, presented. There are RNA companies in South Korea, and so other part of the world. So they, you know, they thought about doing it and they established a company. And so that's what, you know, is if you have ideas and you want to pursue it, you know, you might find some idea and you establish a lab, and maybe, you know, some postdoc from your laboratory can go out and be a CEO. But, you know, that's what I can say, that try to act yourself, not to expect that somebody else will like your idea. It doesn't happen. You have to make.
Thank you very much.
Well, great answers. Now let's move on to the next question. Ms. Emily Yamamoto, are you here?
Yes, yes. Thank you, Dr. Karikó, for a detailed presentation. I'm a high school graduate still preparing for university, and I'm very much interested in gene modification and therapy related with human immunity and the microbiome. Currently still exploring the field, learning from researchers like you. And I'd like to know what are situations when mRNA therapy or mRNA vaccines are preferable compared to therapy using DNA modification and DNA vaccines.
So my colleague here, David Weiner, actually, who was established Inovio, the company who is making a DNA vaccine. And we did experimentally for our urokinase receptor research, we did generated antibody with injecting mice with plasmids, you know, like using as a DNA as a vaccine. What happened is that somehow unproportionally you have to increase the dose with the size of the animal and all the way to human when you use a DNA vaccine. And what was interesting right now, 30 microgram SARS-CoV-2 vaccine needed for a mice, and that's the same for a human. And so what is the reason is that you don't have to upscale, is because every cell can make you know, translate the RNA. And when you inject it, most of the particle will be picked up by immune cells, and they can translate RNA. What happened is that if you transfect the dendritic cells with the DNA, you don't get anything, because for DNA you need cell division. So that's why, you know, that the DNA vaccine is not happening right now. I don't know, are you aware of that? It is in an advanced stage? Not really. And if you want, you know, interesting, you know, when I did the RNA at the beginning, you know, everybody said that, you know, it's not useful because, you know, it degrades. But I also realized when people deliver DNA to a cell, like an epithelial, which half-life was like 5 days, even the DNA therapy is not permanent because the cell is gone. The DNA, what you delivered, is also gone. And what happened is that it seems like even the dream with the gene therapy will be fulfilled with RNA, because now you can see that the companies established and they are starting human trials. Here it's just interrupting gene which makes false and product, but now they are also working on to changing and introducing the correct sequences. So there is and using the gene editing enzyme delivered as an mRNA. So maybe the RNA will fulfill the dream of the DNA gene therapy.
Yes, the RNA has more flexibility compared to DNA.
Because for DNA, if you are working with cells, you know that it had to be, when you seed it and you want to deliver a plasmid, you have to seed the cell subconfluent because the cell had to divide. That's how the DNA can go to the nuclei. But most of the cell in an adult is not dividing. So what happened in the mice, it works. But in a needle track, you have cells which you can transfect very well when you deliver a plasmid. But when you deliver with the needle in a human, it is not 3,000 times bigger like the mice. The human is 3,000 times bigger than the human is 3,000 times bigger than the mice. So, the needle track is not 3,000 times bigger, so that you have some cells, maybe divide there, pick up, but not enough for a full body to have effective vaccine. That's how I see it.
I see. Thank you. Thank you very much.
Okay. Well, let's move on. Ms. Maria Melnui, are you here?
Oh, Maria? That
I think you're muted. Maria, you here? Okay, we just wait and
Maria, actually, I have to tell you that was my classmate in elementary school.
Oh, really?
And now she's working in Japan. She's a physician and scientist there.
Okay.
And yes, we attended it elementary school.
Well, very great. Probably Maria is not really available right now, so we'll move on to the next questions. Mr. Yuta Nakagawa.
Yes, hello.
Go ahead.
Thank you, Dr. Karikó, so much for your inspiring story. My name is Yuta Nakagawa. I'm a PhD student at the University of Tokyo. And in your talk, you kind of mentioned that everybody's talking about the mRNA now because they hear about it in the media and all that. But I'm sure you are quite aware that there are also a lot of false information about these vaccines that are spreading and things like that. So I'm kind of curious that what do you think we can do as researchers to better communicate the findings that we report to the world, and so that there are less false information and things like that? Thanks.
Yeah, so during the pandemic, I learned that we haven't done a good job in the past to explain what a scientist is doing. That when talk to reporters and they ask me that whether I look at the RNA under the microscope, they were not even a reporter who had not knowing that you don't see in a solution something. And so what happened that we did not learn to educate the public.
Yes.
That's what we are doing. And we also, I like to talk to other scientists and exchange and listen and I did not thought about that, try to come up with simple language to explain somebody who is not familiar with my field, that how I explain what I am doing. But we should have done that, because what happened is that we were so busy in the laboratory when the pandemic happened, and those people who were like 20 years ago, maybe was close to some laboratory, they had time and they went to the studio and they were telling stupid things which was not true, but they were just grabbed 5 minutes fame, and so I genuinely believe that there are people genuinely wanted to understand, and we failed them because we were not there to explain that what it is, what we know. Just honestly, we don't know today, and we didn't say openly that we don't know about this virus. It just happened. It could be this or that and that. So, or just saying that we don't know and what we know, and so not to let others to speculate. And we need trustable people, scientists in front of that the public would believe them and trust them that yes, they cannot do everything, they don't know everything, and would give the answer what we know and how, and explain simple language. So that's a lesson for all of us right now, that as a scientist we have to forthcoming and do what can be done, and you know, tell also to the movie industry that they should do more film about scientists, what they are doing, and because what happened, the media is also, I just wonder that if you go out in the street and ask here, you know, that US, and you ask, can you tell a scientist, you know, a name of a scientist, maybe they would say somebody like Einstein, and they would ask, can you tell a scientist who live alive today? They wouldn't able to say. And you know, and why is there? They are taking a pill every day, and their life depends on, and they never ask who come up with this. I have to thank to this person who come up with. They never even ask. And you know, name of people did nothing, only famous because famous. And so the question why the media is not paying more attention to the scientists, and what we should do about it. We have to go out and explain and help. We have to.
Okay, thank you so much.
All right, thank you. Ms. Cherry Yingqi Liu, if I pronounce right, are you here?
Oh, yeah. Dr. Karikó, thank you for an inspiring presentation. I am Cherry, an undergrad at Keio and a researcher at Harvard. And I have two questions for you. First one, if you could elaborate on how lipid nanoparticles were a key part of the vaccine technology. And my second is if you have any advice for rising scientists in exploring different fields. Thank you.
So the lipid nanoparticle is, you know, not just inert, not just something that protect the RNA. Yes, of course, it had to be protected from the degradation when it injected to the body, and when it enters to, you know, helping the RNA to enter to the cell, and when it enters in the endosome of these immune cells. Now that they had to release the RNA, and then the RNA had to escape. So we are asking so many things from this lipid nanoparticle, but more importantly, one component, as we just recently published, as is the ionizable lipid, is the adjuvant in the vaccine. So the RNA is non-immunogenic, and of course we know that we need an adjuvant, and this is the ionizable lipid. And so this is the role of this LNP. And what was the other question you had?
Maybe she's offline right now. I can see her name in the screen.
Okay.
All right. I have several questions on the chat. One question is that has anything been done to make the mRNA vaccine more effective and long-lasting?
I mean, you know, the long lasting is depends on very much the induced immunity. We could see that as we learned during the pandemic, that people who get infected, we learned that 6 months later they could be reinfected. So we know that against this virus, it was the protection was not permanent. It was not for a lifetime, and so it is maybe the nature of it. I am not a vaccinologist, and not you know, not an immunologist either. I am a chemist by training, so that's what you know. In these days, everybody is an epidemiologist, everybody is a vaccine expert, but that's what my understanding is. So that it would be good to have a vaccine which would be protecting from against all of this variant. My understanding is that the more vaccinating with the same mRNA coding for the same protein repeatedly is it not only increased the level of antibody, but it is the breadth of it. So it is the diversity of antibody increase. So then this is the reason why after the third vaccine injection that it could the antibody generated in the patient in the people could neutralize the Omicron as well.
Okay, thank you very much. Maria and Councillor Hosu has given us the chat. Basically the same question. When will you come and visit us in Japan? And thank you for the lecture. Do you have plans to visit Japan soon?
So yeah, I mentioned that maybe in April I will visit Japan the third time now, and I'm looking forward.
Yeah, thank you so much. Well, there should be a lot of questions, but unfortunately we are reaching the limit of the time, and I have to close my session. It was my great honor to chair and listen to Professor Karikó's wonderful lecture today. Learning the years of story behind the scientific career of Professor Karikó, and of course the discovery of COVID-19 vaccine, I'm once again so grateful that we had such a great vaccine in our hands soon after the pandemic began. And alas, as a physician who confronting the COVID-19 patients these days, I see that the vaccine not only gave us the protection, but it really gave us the strength or the courage to go through all the devastating situation, which let us remind that we're not alone in the science and the scientists, including Professor Karikó, are with us. Yeah. And again, it was a very, very great pleasure to have Professor Katalin Karikó, the laureate of the 26th Keio Medical Science Prize. And I would like to once again thank Professor Karikó for giving us a remarkable lecture. And thank you very much for your participation. Thank you so much.
Thank you.